Today, we'll be talking about the absorption and metabolism of vitamin D. So if you haven't subscribed to this YouTube channel, please do well to subscribe. Just click on the subscribe icon. As well, click on the notification icon so you can be notified whenever a new video is being uploaded. Also, please do well to like and share this video. So, vitamin D occurs in two forms. We have the vitamin D2 and then we have the vitamin D3. Now, vitamin D2 is called the hegocalciferol, while vitamin D3 is called the cholecalciferol. So, these vitamins have their respective precursors. So, the precursor of vitamin D2 is called the hegosterol. That's vitamin D2 precursor. It's called the hegosterol. And the hegosterol is usually found in plants. So it's usually found in plants, but it can also be found in the cell membrane of fungi and protozoa. So the cell membrane of fungi and protozoa. But then, because this is the precursor form, it is inactive. And so when they are obtained in diet through the consumption of plants, they are not well absorbed because they are inactive. Now, the inactive form of vitamin D3 is dihydrocholesterol. Dihydrocholesterol is the precursor form of cholecalciferol, and this is also the inactive form. Now, this dihydrocholesterol is usually obtained from cholesterol. So this is obtained from cholesterol and it occurs in the liver. Okay, so cholesterol is converted to dihydrocholesterol in the liver or in the intestinal mucosa. Then after it is formed, it is now transported to the skin where it remains. So now in the skin, it gets exposed to UV light, okay, like the solar, and then it becomes converted. It undergoes an intramolecular activation or photochemical activation to become activated or become active. The active form is now called the cholecalciferol. Also, the egocalciferol, which is the precursor, which is inactive, when it's exposed to UV light, it becomes active. It is converted to egocalciferol and becomes active. Now, this egocalciferol, when taken in from plants, is well absorbed. Now, there are several dietary sources of vitamin D. For example, we have the fish liver oil, we have the egg yolk, the margarine, butter, cheese, and several others. Now, when vitamin is taken in, whether it is in vitamin D2 form or it is in vitamin D3 form, it is usually absorbed in the small intestine, specifically in the duodenum or in the jejunum. Now, its absorption is enhanced by bile salts. So bile salts act on large fat droplets. That is because vitamin D is fat soluble. It is taken in from the diet combined with fat. So in the small intestine, bile salts act as detergents and hydrolyzes the large fat droplets into smaller ones. And by so doing, it gives room for lipase to also act on those smaller fat droplets. And by so doing, it gives room for vitamin D to be free and then be absorbed. Now, when vitamin D is absorbed into the circulation, it binds to vitamin D binding protein. Vitamin D binding protein. It can also get bound to albumin. Now, when this binding occurs, it is transported to the liver. It is transported to the liver. Now, in the liver, vitamin D undergoes a process of hydrolysation where an hydroxy group is being attached to it, and that hydrolysation occurs at position 25 of the vitamin D structure. Position 25 of the vitamin D structure, and that hydrolysation will now result in 25 hydroxy vitamin D. Okay, this can also be written as 25 hydroxy vitamin D. You can say vitamin D. Often it is usually written in this form, but because it can be vitamin D2 or vitamin D3, let's leave it at this. So now, this process of hydrolysation is catalyzed by an enzyme called 25 hydroxylase. So this is the enzyme that catalyzes the hydrolysation of vitamin D in the liver to form 25 hydroxy vitamin D. Now, after this 25 hydroxy vitamin D is formed, it is also released into the circulation, where it also binds to, of course, serum globulin, and then it is carried to the kidney. In the kidney, it also undergoes a process of hydrolysation, but this time the process of hydrolysation occurs in position 1. So that will now result in 125 dihydroxy vitamin D. 125 dihydroxy vitamin D. Now, the formation of this is catalyzed by an enzyme called 1-alpha hydroxylase. 1-alpha hydroxylase. Now, this 25 hydroxy vitamin D that was formed in the liver can also be called calcidiol. Whereas this 125 dihydroxy vitamin D is also referred to as calcitriol. This 25 hydroxy vitamin D is the main form of vitamin D present in the circulation. So, when vitamin D is being assayed for, it is actually in this form that it is being assayed. However, 125 dihydroxy vitamin D is the active form. So this is not the active form. This is the active form of vitamin D, but this is the form of vitamin D that is present in the circulation. So whether the hydrolysation occurs in the liver or in the kidney, it must occur in the presence of certain cofactors, which include the magnesium ion, the reduced form of nicotinamide adenine dinucleotide phosphate, and then molecular oxygen. The functions of the calcitriol includes 1. It enhances the absorption of calcium and phosphate from our diet in the intestine. Okay, and also it enhances reabsorption of calcium and phosphate from the renal tubule back into the circulation. and then it also stimulates the release of calcium from skeletal system into the circulation. Now, for every activity that goes on in the body, there must be some form of regulation to ensure that biochemical components are not produced in excess, or certain activity are not being over exaggerated, and that is a process of what? Regulation. So the production of calcidiol and calcitriol has to be regulated. So for calcidiol to be regulated, there will be a negative feedback inhibition on the enzyme that catalyzes its production. So if sufficient amount of calcidiol has been produced, there will be a negative feedback inhibition on this enzyme that catalyzes its production. So when this enzyme is being inhibited, then the production of calcidiol will stop. Also, for calcitriol, there will be a negative feedback inhibition on 1-alpha hydroxylase. To add to it, apart from the negative feedback inhibition, the presence of parathyroid hormone can also regulate the production of calcitriol. Also, the presence of serum phosphate level can also regulate the production of calcitriol. Of course, in a state of hypocalcemia, there is usually an upregulation or an increased production of parathyroid hormone from the parathyroid gland. Now, this increased production of parathyroid hormone stimulates an increased activity of 1-alpha hydroxylase. And when the activity of this enzyme is being increased, there will be an increased production of calcitriol. An increased production of calcitriol will result in an increased production of calcium. On the other hand, an increased level of phosphate in the circulation will result in a decreased production of calcium. That is because 1-alpha hydroxylase will be inhibited. So an increased concentration of phosphate level in the circulation will inhibit 1-alpha hydroxylase, which will result in the decreased production of calcitriol, which will in turn result in a decreased production of calcium. So in my next class, we'll be discussing the functions in detail, the function of calcitriol, and then we are going to talk about the deficiency as well as the excess of vitamin D.